project, the work that I'm talking about today was like, it spanned the course of about 5 years. It spanned the time while I was at Stanford and the time while I was at the Broad Institute. So there's sort of a mixture of different types of analyses that went into this paper over the course of that quite long project span. So should I go ahead and just jump into it, Dennis?
Sure.
Okay, great. So just to get started, the main premise for doing these kinds of studies is that genomics is vastly biased towards European participants. In 2009, there was a study showing that 96% of participants in GWAS were of European descent. Just last year, this was updated to show that now we have about 81% of European participants in GWAS studies. So we're making some progress in increasing diversity. But even of that 19% that's non-European, the vast majority is of East Asian descent. So there's this huge swath of genetic and phenotypic diversity that remains unqueried in genetic studies right now. Of course, as you can plainly see with your eyes, and in disease biology, there's an enormous amount of genetic and phenotypic variation across human diversity, but our genetic studies currently miss the breadth. So two very visually obvious phenotypes that are quite variable that we were interested in here for evolutionary reasons were skin pigmentation and eye color. We've focused throughout the study pretty much on skin pigmentation and tanning. We studied individuals from Southern Africa, so these Khoisan populations. We were particularly interested in these populations because they form an extant lineage in modern human populations. So they're among the most diverse. They have rapid linkage disequilibrium decay compared to other African populations. So all of these features indicate that the Khoisan populations of Southern Africa form one of the earliest populations in the modern human lineage. And this picture on the bottom right shows 3 of the study participants from our work that live in a household. And you can see that within a single household, there's an enormous amount of variation just in skin color. Prior anthropologists have gone around over the course of time and have tried to get representative numbers for skin color across different populations. And if you look at this map from Renato Biasutti in 1959, what you can see is that there's an enormous amount of variability in pigmentation, especially in Africa, which spans the equator and has quite a bit of latitudinal variation. So that indicates, because of this enormous phenotypic variability, that Africa would be an especially useful place to study skin pigmentation. There's opposing selective forces that have been proposed to act on skin pigmentation. The best studied is vitamin D. So as you move to higher and higher latitudes or further and further from the equator, it's been pretty well established that skin pigmentation should likely lighten in order to increase vitamin D production, which helps stave off severe diseases such as rickets. On the other hand, it's a little less clear why skin should darken, but there are several hypotheses as to why that might be. One is skin cancer. So skin cancer typically affects individuals post-reproductively, so that may not make as much sense in an evolutionary context. But I think what's has a little more grounding is protecting folate, which is very important for development. When women are pregnant, if they get inadequate amounts of folate, there are more likely to be birth defects. So protecting this from photolysis is important close to the equator. So that's one hypothesis for why skin should be darker. So I showed you this map of how variable skin pigmentation is across the globe, but what I didn't show you is where skin pigmentation studies had been done when we started this study. So if you look at where all these little dots are, this is just where skin pigmentation association studies were performed when we set out to do this work. And you can see that Africa is completely ignored in all association study efforts, leaving it as very low-hanging fruit for skin pigmentation work. So I think this indicates that there's an enormous opportunity here that's just been completely missing in prior studies for various reasons. But it's high time to start doing association analyses in Africa. So one of the first things we wanted to look at is how much skin pigmentation varies across the globe. So, we had some nice data from this anthropologist who was going around in the late 1950s, but we wanted to update this with some of the very large studies that have taken place since then. So fortunately for me, this community is really great to work with. I've reached out to a number of different colleagues in the field and asked if they would be willing to share phenotype data from these different locations indicated on the map. And over the course of time, we were able to collect about 5,000 phenotypes representing about 33 populations across the globe. So when we order these different populations by latitude and look at the distribution of this M index, I should mention what the M index is. Basically, if you want to measure skin pigmentation, an easy way to do that is to use a spectrophotometer, which just measures the amount of light reflectance. So for the M index, this is measuring a log scale of the fraction of red light reflectance. So the darker your skin, the higher the M index. So you can see on the x-axis on this right plot that the M index spans from about 25 to 125. And if you look at the European populations at the highest latitudes, you can see that these distributions don't vary much at all. So they're pretty narrow. And as you get closer and closer to the equator, for example, with these gray Melanesian populations or this green Ghanaian population, you can see that the variance starts to increase. So the two populations that we studied for this work were the Nama and Kamani-san. Those are highlighted in this red box, and you can see that there's actually more variability given their latitude than you might expect. And part of that comes from admixture, which we'll talk about a little bit more. You can see in this orange distribution, the Cape Verdeans in an island off of West Africa, which has about 50% European ancestry and about 50% West African ancestry. This population varies quite a bit in skin color as well, and that's also due to this high rate of admixture. So as I mentioned, recent admixture has a quite strong effect on pigmentation. So on this top panel in A, basically I'm just giving you a breakdown of what ancestry looks like across these different populations. So each line here, this whole column, indicates an individual. And here I've broken this down into 7 different populations. So on the right, you can see the CEU, or these Europeans from Utah, and the French populations in blue. You can see this green color corresponding to West African ancestry, this Yoruban ancestry, or Bantu ancestry. In the brown color, you can see for the Sandawe and Maasai, these are representing some East African ancestry. This yellow Hadza component is indicating this ancestry from this other hunter-gatherer group. And then in the red, you're starting to see this Khoisan-specific ancestry. So the red is indicating, which through some work that we did in another paper, we can see that this is sort of indicating this northern Khoisan ancestry or northern Kalahari ancestry. And in the orange is indicating some southern Kalahari ancestry. So if we pull apart these ancestry components and do an association analysis with skin color, what you can see is pretty much what you would expect. So in this panel on the bottom left, this Bantu ancestry, this West African ancestry, having more West African ancestry increases skin color darkness, or increases this M index. That seems pretty sensible. Having more West African ancestry makes your skin darker is basically the takeaway there. Conversely, having more European ancestry makes your skin a bit lighter. Kind of interestingly, having more Khoisan ancestry neither lightens nor darkens skin. So it just seems like through a few different approaches that this Khoisan ancestry seems to increase the variability in skin color. So one aspect of this study that was particularly challenging in some statistical analyses comes from the fact that sampling in small indigenous communities results in some biases. So we had a translator helping us go around in the field and helping us connect with the community and act as a liaison, both as a translator and as someone who could help us identify individuals who might be interested in working with us. I should mention that doing these sorts of sample collections requires an enormous amount of community engagement. Of course, it requires you to meet with community leaders to ensure that this work is ethical, that the community agrees to participate, despite the fact that there may be no immediate reward for this. We also make sure that we get some in-depth anthropological information. So when we sit down with individuals to do this kinds of phenotyping, we also ask which languages do you speak, which ethnicities do you self-identify with? And we ask this about both the individual themselves, their parents, and then their parents' parents. So we get 2 generations worth of information through these anthropological interviews and through these ethnographic interviews. We measured skin pigmentation. We also measured wrist pigmentation so that we could subtract the sun-exposed wrist area from the underarm skin pigmentation, which, since it's not sun-exposed, gives you a rough proxy for the baseline level skin pigmentation. And then we also measured height and collected samples they had to spit in a tube until it reached a certain level, which is sometimes a bit of a challenge. So as I was mentioning, doing sampling in these small indigenous communities leads to biases because your translator is more likely to take you to visit their cousin and to visit their aunt and to visit their parents. So you end up with these highly connected pedigrees, and having this detailed level of ethnographic information means that you can construct these multi-generational pedigrees, but also means you have fewer unrelated individuals. But that's kind of true in general in these kinds of communities. There's typically a bit more pedigree structure than you would find in a cosmopolitan population. So based on the fact that we have genetic data from SNP arrays, and we also have exome sequencing data for a subset of individuals, and we also have pedigree information, and there's a bit more relatedness than typical, we can actually assess heritability in several different ways. So we used the standard approach of GCTA to assess baseline skin pigmentation heritability. We estimate that pigmentation has a heritability of around 0.9. In contrast, tanning status, so basically your underarm pigmentation minus your wrist pigmentation, or the reverse of that, you see that heritability is about 0.3, but this is not significant, and we have fairly small numbers for this analysis. I should mention that because there's quite a bit of relatedness in this particular sample set, that this estimate is more similar to what you would find from a family-based study than what you would find from a typical unrelated SNP-based heritability estimate. So if you look at the bottom row of this table, you can see that when we estimate heritability from using just the pedigrees and no genetic information whatsoever, that we get fairly consistent estimates as with the SNP-based heritability. So here we estimate that baseline pigmentation has a heritability of around 0.96 with a standard error of 0.12. In tanning status, in contrast, does not seem to be significant. We're estimating it to have a heritability of about 0.19 with a standard error of 0.11. So skin pigmentation here, through various different methods, we estimate very consistent heritability around 0.9 to 0.97. In tanning status, through various different approaches, doesn't necessarily seem to be heritable. This is sensible based on how you tan. This is primarily environmental. How much time you spend outside is highly correlated with tanning status. So the next thing that we wanted to do was take what people know about pigmentation. There's been a ton of work on pigmentation already in different populations, and there are quite a few genes that are known to have varying ranges of effects, quite larger effects than in your typical standard GWAS analysis of a complex trait. So for example, SLC24A5 is the best studied pigmentation gene, and it's known to have one of the largest effects on skin color, and is thought to have evolved in Europe and to lighten skin color. So we took 3 candidate gene sets from previous studies and asked how much of the heritable variation can be explained by these different gene sets. So we partitioned this into several different groups. So when we looked at the first gene set, this is the smallest gene set that was the second smallest, that was based on prior association. So here we have about 15 genes that are known from previous association analyses to be directly affecting skin pigmentation in human populations. The second candidate gene set is a gene set that's a little bit larger. It consists of about 50 genes that are based on functional and molecular evidence. So these can be associated with skin color in humans, but they can also come from animal studies. And then the third gene set came from our association analysis. So if we look at how much of the heritable variation each of these gene sets can explain, gene set 1 can explain, 10 to 15% of the heritable variation, but this is not significant. So this, could be 0. If you look at gene set 2, this is also not significantly different from 0. This explains about 12% of the variation as a, point estimate. If you look at gene set 3, this is starting to explain a bit more of the heritable variation. This explains maybe about 23% of the heritable variation. That's, maybe that's what we would expect, given that we're doing an association analysis of pigmentation in the same population as we're measuring it in. So these results indicate that there's a huge amount of genetic diversity contributing to pigmentation that we don't understand yet. But that's not to say that these gene sets don't matter. So if we look at those gene sets from previous studies and ask how much of the variation do they explain compared to a random set of genes, in both cases, they're explaining a significant amount more than a matched gene set. So, matched on number of exons, matched on gene size, and those sorts of things. So these candidate gene sets are important, but they don't explain a substantial fraction of the heritable variation in the Poisson specifically. So the next thing that we wanted to try to get at was sort of a conceptual overview based on the phenotype analyses and the genetic analyses. If we know that a little bit about selection from prior studies, and we know about the fraction of heritable variation that can be explained by candidate genes, and we know a bit about their effect sizes, can we put together sort of a model for how darker skin evolved? So what we think is going on in this first model is that there are differences in selective pressures. So it's been shown time and time again that convergent evolution in different populations that live far from the equator have undergone directional selection. So European populations, for example, have a few very large effect variants that are known to contribute to pigmentation, and those have swept to high frequencies, or in some cases to fixation, to confer this light skin color. The same has happened in East Asian populations, sometimes with the same genes and sometimes with different genes. So there seems to be, at high latitudes, strong effects of directional selection. For darker populations, we think that what's happening is that there's actually some stabilizing selection. So maybe there's also some directional selection at play, but it seems like there's sort of like a fitness optimum where having darker skin is beneficial close to the equator, but it's not just that having darker skin is important, otherwise you would be pushed in one direction and there would also be relatively large effects and some fixed effects. But what seems to be the case here is that there are sort of a variety of different SNPs that are favorable that contribute to dark skin close to the equator, and that it's useful to have some variability in dark skin. So perhaps there's some stabilizing selection increasing the variability of the phenotype here, and then intermediate is somewhere in between, of course. And then a second model for what might be happening, which is not mutually exclusive, is that perhaps the target, the mutational target across the entire genome for contributing to light skin is a little bit smaller, and there are fewer loci contributing. So the causal effect sizes for light skin tend to be larger, and perhaps the causal effect sizes for darker skin tend to be smaller, and there are more of them. So we think that both of these are sort of at play and are contributing to this large role of polygenicity, specifically in darker skin. We also looked at how divergent different genes that contribute to skin color are between the Poisson populations from Southern Africa and Europeans and West Africans. So in both of these cases, if we look across the entire genome, I'm showing you in A and B what the distribution of this FST or fixation index are. So an FST of 0 means that the SNPs or this particular region of the genome is completely the same between the populations. An FST of 1 would indicate that they are completely different. So one is fixed for, say, the light skin allele, and one is fixed for the dark skin allele. What you can see through this distribution is that there is greater divergence between the Southern African population and European population on average. That's sensible based on the fact that Poisson populations diverged earlier in time from European populations than from West African populations. And if we superimpose the pigmentation genes on top of this distribution, what you can see is that there's a few genes that stand out or are huge outliers on these distributions in both populations. So for example, SLC24A5, which is canonically known to be one of the largest effect pigmentation genes, it's been very well studied, is a bit of an outlier there, as is OCA2 in this FERC2 region. So I already mentioned a bit about SLC24A5, and you can see the zoom in of this FST over this region for both the comparisons between, the pairwise comparisons between the Poisson in West African and the Poisson in European populations. OCA2, I think, is sort of interesting because it's known to have the largest effects on eye color of any gene that's been studied in the human genome. Interestingly, in Europeans, this contributes to blue eye color and green eye color and brown eye color. The Poisson actually also have quite a bit of variability in eye color. I haven't really seen too many blue eyes, but there's certainly a lot of green eyes and hazel eyes in the population. We don't have quantitative metrics of eye color, but I think it's interesting to note that there's quite a bit of divergence here, indicating that perhaps some evolution that's unique to the Poisson has been at play here. So in the last little bit, I'll just tell you about the association study that we did. So it was a complex study because of the time that passed and the number of sample collections that went on. But there were two phases. So in the first phase, we genotyped a couple hundred individuals split between the Kumani and the Nama. And then we took that first phase of association analyses to highlight our favorite top 35 loci, captured those and did targeted resequencing later on the entire sample. In the second phase, we did the same thing with about, again, a couple hundred individuals. So in total, our association analysis had quantitative phenotypes and association summary statistics for about 430 individuals. And then we did targeted resequencing on the same set of individuals. So from the association analysis using the meta-analyzed data, this is what our association analysis looks like. You can see this Manhattan plot on the left and a QQ plot on the right. This QQ plot is indicating that there's some liftoff, from what you would expect, indicating that there is some true signal here. If we look at the Manhattan plot, we can see that our strongest signal comes from SLC24A5. This is a canonically well-studied pigmentation gene. And there's a couple other genes that are pretty sensible here. So TERP1 is not meeting GWAS significance, but is known to impact albinism. In humans, it's also known to contribute to blonde hair in Solomon Islanders in Melanesia. There's another gene that's not meeting GWAS significance, but looks like it has a LD peak that's approaching significance. This is SNX13. This has not been identified in the context of pigmentation before, but there are some burgeoning studies indicating that it, may play a role here. When we did our targeted resequencing, the slide didn't turn out very well, sorry. In panel A, you can see our QQ plot there. So, of course, SLC24A5 pops out again. And then there are a couple of other genes that are interesting. SMARCA2 is particularly interesting because it's been hypothesized to have a role in pigmentation in model organism studies before. It plays a role in chromatin activation at specifically loci where MIDF is recruited. MIDF is termed the transcription factor that's the master regulator of melanogenesis. And that was particularly interesting because we found multiple independent signals between SMARCA2 and VLDLR. So we think it would be interesting to try to follow up on these. So just some conclusions from there. Skin pigmentation is highly heritable and variable with intermediate skin observed in Southern Africa, and I think this has been underappreciated in the literature so far. So there's quite a bit of low-hanging fruit for skin pigmentation studies to undertake. Candidate genes from prior human skin pigmentation studies and from model organism studies explain a small fraction of the heritability in Poisson populations specifically. And skin pigmentation is very polygenic. There are more associations closer to the equator and fewer at high latitudes, indicating that we really don't know what makes darker skin darker. And I think there's a point to be highlighted across this type of work in general, in that greater diversity is needed in these genotype-phenotype studies to understand how humans evolved. And it's especially important to take these into account when there's such vast differences in the environment that selection is acting on. So here, far from the equator, it's important to keep in mind that there's an enormous amount of genotype-phenotype variation here. So just as a few acknowledgements, my collaborators here have been great and have spanned quite a broad geographical spectrum. So I have collaborators to thank at Stanford for sure. So Carlos Bustamante was my PhD advisor who helped spearhead a lot of this work. Brenna Henn was the senior author in this work. She was a postdoc while I was at Stanford, and she's has been super great as an anthropologist to work with. We have a bunch of collaborators in South Africa at Stellenbosch University that were integral to doing a lot of the anthropological and on-the-field work. The Khoisan community in general was incredibly amazing to work with. Without them, this work definitely would not have been possible. And of course, MGH, Mark Daly, my postdoc advisor, has been very helpful in getting this across the finish line, and then my funding, of course. And thank you guys for listening, and I'm happy to take any questions from the few of you who might be here.